This is a lesson for year 12s and year 13s for your A-level biology. It's also useful for any year 11s thinking about going into doing A-level biology. Last time we looked at the light-dependent reaction. Today we're looking at the light-independent reaction. And the question I'm thinking about is, can photosynthesis happen in the dark? So, recall quiz though, quick high five. Number one, what process splits water? Number two, what are the products of this when water is split? Number three, what happens to electrons when light is absorbed in the chlorophyll molecule? And what are the products of the light-dependent reaction? And number five, what are they needed for? So there's your 5 questions. Press pause, have a go answer those questions, and when you're ready for the answers, you can unpause the video. Cool, right, so if you've done that, so number one, what process splits light? It's called photolysis or photolysis. Photolysis. What are the products? Well, you get hydrogen ions, you get electrons, and you get oxygen. Okay, so you get hydrogen ions, electrons, and you get oxygen as a byproduct. What happens to electrons when light is absorbed? The electrons become excited. They raise energy levels. What are the products of LDR? So eventually the products you get from the light-dependent reaction is ATP, and you get reduced NADP, or also known as NADPH. You also get oxygen as the byproduct. What are they needed for? Well, oxygen is a byproduct. It's not needed directly, but it does get used for respiration. But any excess is passed into the atmosphere as a waste product. But the ATP and the reduced NADP, the reduced NADP is what's happening in today's lesson then. So that's what they're used for. They're used for the light-independent reaction. But how? What do you need to know? You need to know, explain how carbon dioxide absorbed by plants is incorporated into inorganic molecules. So what's the carbon dioxide for basically in photosynthesis? Describe the roles of ATP and reduced NADP. What we just talked about there, the products you get from the light-dependent reaction. They enter the light-independent reaction, what we're talking today, and how they're used. Describe the events in the Calvin cycle, which is what those two things are involved in. So last time we looked at light dependent because it depends on light. Light is needed. So obviously this means light independent. It means this doesn't need light. So this part of photosynthesis doesn't require any light. Does that mean photosynthesis can happen in the dark? The answer to that question is obviously no. You can't get photosynthesis happening in the dark. This bit can happen in the dark, but photosynthesis as a whole can't, because if you don't get the products from the first stage, which was the light-dependent reaction, if the LDR doesn't happen, the LIR can't happen. Okay, so what does it look like then? It's known as the Calvin cycle. So this is the Calvin cycle, named after a scientist called Melvin Calvin, and we'll talk about him towards the end of this lesson. So where should we start? Well, remember we've got reduced NADP from our light-dependent reaction, and we've also got ATP from our light-dependent reaction. So it's like dotted off from that corner over there. That's last lesson. Those two guys enter our system. Okay, so where shall we start? Now the Calvin cycle is a cycle, so there's no beginning, there's no end, but we need to start somewhere. So we'll start off here with number one. Here is our carbon dioxide molecule. So there's the CO2, carbon dioxide molecule. Now, you've got this compound called ribulose bisphosphate. And if you notice, it's a 5-carbon compound. So it's an organic compound, and it's got 5 carbons in there. So ribulose bisphosphate. Now, there's an enzyme called ribisco, or a coenzyme, ribisco, that's what the coenzyme is, that is needed to join the carbon dioxide to ribulose bisphosphate. Now, if you get 1 carbon plus 5 carbons, how many do you get? Well, it's kind of obvious. You're going to get 5 plus 1 is equal to 6. So you're going to get a 6-carbon molecule here at stage 2 here. So what happens to this 6-carbon molecule? This 6-carbon molecule then gets split. It gets split into 2 3-carbon molecules. those 3 carbon molecules are called glycerate 3-phosphate. Glycerate 3-phosphate. Now you can, once you've written the whole word out in an exam, you can simplify it to 3GP. We call it 3GP. So the GP stands for glycerate phosphate. We use the 3 to represent it's got 3 carbons in it. But you're going to get 2 lots of them, because remember you've got 6 carbons, because they're joined together here. So that gets split into 2 lots of 3. So this 3GP. The 3GP then, this is where the first bit, where the ATP, okay, from our light dependent reaction is needed to help convert. So ATP is transferring energy to convert, and also our reduced NADP is becoming oxidized. It's becoming oxidized because it's lost the hydrogen ions. The reduced NADP becomes oxidized, therefore the 3GP is being reduced. That must be gaining the hydrogen ion. So this is being reduced, it's also gaining some energy, and this is converting the 3GP into triose phosphate. Now this is a really important compound, triose phosphate, also known as TP. So you got 2 lots of TP. Now here's the thing, I've got 2 lots of 3, but I'm trying to make glucose. Now if you know what glucose, the formula for glucose is C6H12O6. So C6, there's got to be 6 carbons in there. Now how's that going to make it? It sounds like, oh well, why don't I just stick those 2 together and I'll get my glucose molecule. It's not as simple as that, because it's a cycle. This stuff has to keep getting recycled over and over and over again. So I need this to go back to the beginning. Well, the beginning was a 5 carbon compound. So if there's a 5 carbon compound here, the ribulose bisphosphate, I've got a 3 and a 3. What needs to happen is that one of those carbons needs to come off this little conveyor belt. I give you like an analogy. It's a bit like coming back from the airport and you're trying to collect your bags. Obviously you're not doing that now because all the airplanes are grounded. But when you go around, you're looking for your bag, and the bags go round and round and round the conveyor belt. So you imagine you put your bags on the conveyor belt, it comes back around, and you drop one off, okay? And then another bag, and you drop one off. Now, if you go round 6 times, every time this one carbon jumps on, from your 2 lots of TPs, if you take one off, there, one carbon comes off, you're going to get your 3 and your 2, they're going to join back together again. So when they join back together again, that's what's happening here. And it needs energy. So you got another ATP turning into ADP plus PI, so energy is being transferred to turn it back into ribulose bisphosphate. One of the carbons has been dropped off from that conveyor belt, your suitcase, you picked it up. Now, therefore, you want to make 6 carbon compounds. How many turns are you going to need of this Calvin cycle to end up with 6 spare carbons? Well, obviously you're going to need 6 turns. So for every 6 turns, you're going to end up with 6 spare carbons. So if you think about it though, if I do 3 turns, if I got 3 carbons, I can make one of these extra. I can make a TP. And it's these TPs that I'm trying to make, these triose phosphates. So 3 turns, I get a spare triose phosphate made. Another 3 turns, I get another triose phosphates made. And that's where this useful organic substances comes about, where you get, e.g. your glucose. So you can make sugars, okay? So you can make your useful sugars like glucose. And that is the Calvin cycle. That is the light independent reaction. But how did they come across this idea? How did they come across it? Well, giving you some clips. This one's a bit more complicated, giving you the sort of the atoms, the individual atoms there, showing you what ribulose bisphosphate looks like. This is a cycle going the opposite direction to the one I just showed you. That's your RUBP. So RUBP, ribulose bisphosphate, joins on with this carbon molecule. So then it splits it into 2 lots of 3 carbon molecules, your 3 GPs. You've got ATP there, and your NADP, H, your reduced NADP. That's going to turn it into your TP. Okay, your GPs are going to turn into your GPs, and then your TPs are going to turn back into your ribulose bisphosphate. Now, where does it start from? So carbon dioxide diffuses in, it's dissolved in water into the stroma of the chloroplast. The carbon dioxide then combines with your 5-carbon RUBP. You end up making 2 GPs, your 3 carbon molecules. Your ATP and your reduced NADP, which is your NADPH, from your light-dependent reaction reduces GP to become TP. NADP, then, because remember we just lost the hydrogen there, that actually goes back to where it came from. It goes back to the light-dependent reaction to accept more hydrogen ions, so therefore it can come back to the light-independent reaction. Some of the TP is converted into useful organic compounds like glucose. Most of the TP goes back to make RUBP using ATP from the light-dependent reaction. That's why you need those protons keep going through, so you can make lots of ATP, because you need lots of ATP for your Calvin cycle, because you're going to need 6 turns just to get 1 glucose molecules out of it. Okay, so where does it happen? It happens in the stroma. Remember, the light-dependent reaction happens in the thylakoids, thylakoid membranes, because that's where the chlorophyll molecules are in the transmembrane proteins. Whereas here, the light-independent reaction is happening in the stroma of the chloroplasts. That's where the enzymes to reduce carbon dioxide in the light-independent reaction are. The stroma is fluid surrounded, that surrounds the guarana, so the products of the light-independent reaction can diffuse readily there. And then it also contains DNA and ribosomes, again, to manufacture lots of proteins for and enzymes for the light-independent reaction. Lollipop. How can we use a lollipop to work out the light-independent reaction? Is this a riddle, or am I offering you lollipops? This is also known as the lollipop investigation. This is how Melvin Calvin came across. He actually won the Nobel Prize. A little story then. Melvin Calvin was working on this, trying to find out, if you think about photosynthesis as one of, or if not the most important chemical reactions happening on Earth for life to exist on Earth, because it's the beginning of all food chains, when you've got plants at the beginning of the food chain. Because we call them producers, Melvin Calvin was trying to find out, well, what is happening in photosynthesis? What are the chemicals? What is the biochemistry of this? And he had a student working with him, or a graduate who was doing his PhD, who was becoming a doctor in his own right. So he was doing a lot of the work with him. His name was Benson. So it was Melvin Calvin and Benson. And what happened is Benson, his protege, was doing most of the work probably, and doing all the dog body work, collecting all the data and doing all the hard work with him. And when the Nobel Prize was awarded to Melvin Calvin for his amazing work, instead of sharing the accolade with somebody who did, probably half the work, if not more, with him, he pretty much took all the glory and made it out to be, it was just him. Stabbed his buddy in the back. Very nice Calvin, Melvin Calvin. Right. So we don't try to forget the name Benson. It's one of those horrible stories that you hear about in science. Now, hopefully science isn't, most scientists don't do horrible things like that. We try and do science to, make humanity and civilization a better place. Right, so what did he do? I didn't get a picture of the image of that. If you Google the Lollipop experiment and look at the photos, I should have put one up, you would see what it looks like. It's not a sphere, it's not a glass ball, but more like a flat disc. Okay, so more like a lollipop. What you've got is it's glass, and it's obviously round, but it's not spherical. It's not like a ball shape. It's flat. Okay, so if you imagine you're looking at that, because when you look at that picture, to me it looks more like, because it's got that sort of circle shape in there, it makes it look like it's a sphere. It's more of a disc shape, but it's glass all the way through, and you had light shining on it from front and back. And inside this glass flask, he had algae. Algae are photosynthetic organisms, and he had a nutrient medium, so he had all the right nutrients. You had the water and the minerals and all sorts of stuff you needed for the algae to survive. And he was pumping in carbon dioxide and air, and he also injected radioisotope, so carbon dioxide in the form we had carbon-14. This is radioisotope. So you got, if you imagine sodium hydrogen carbonate, if you add it to water, baking soda basically, it dissociates. You end up getting carbon dioxide dissolved in that water. Now the thing is, you can't see carbon dioxide. Where is it? It's gone. It's a gas somewhere. Where is it gone? I can't see it. I can't follow it. But if you, instead of using normal carbon-12, you use carbon-14, which is radioactive, you can use films that get exposed, and you can follow it and find out what compound it is. And that's basically what they did. So they injected it with carbon-14 as hydrogen carbonate. When it enters the water, it becomes carbon dioxide. But with carbon dioxide, a radioactive version of carbon dioxide. So then I can follow it. I can see what compound has it become. So they injected that, and every 5 seconds, they opened the tap, and the algae with the carbon dioxide inside the algae, obviously, would have fallen into this hot methanol. Now the hot methanol is just hot alcohol, would instantly kill the algae. And what that would do is take a snapshot in time. It's like taking a photograph every 5 seconds. So from 0, 5, 10, 15, 20, 25, 30, 35. You're taking a snapshot. You could do it every second if you wanted to. Okay, but you're constantly just moving that away and then testing what's in there, and then putting another fresh test tube in there, another 5 seconds, and so on and so on. And what they discovered is this. So obviously photosynthesis is happening with normal carbon dioxide all the time, but you can't see where that carbon dioxide has gone. I can't test it, I can't visualize it. So what they did is, by having the radioactive version, they can follow it and see what compound it was. So at 0, that carbon dioxide, it was carbon dioxide, because it hadn't been absorbed by the algae and hadn't been processed in photosynthesis. So at 0, it was just carbon dioxide. Then, after 5 seconds, there's no carbon dioxide. It was now GP. After 10 seconds, there was some GP and there was some TP. After 15 seconds, there was GP, TP, and glucose. And after 20 seconds, there was GP, TP, glucose, ribulose bisphosphate. After 25 seconds, it would have stayed the same as that all the time. Okay? And then they said, right, okay, that must be the end of the cycle. So where does it all start? And that's how they came up with this, the Calvin cycle. So algae grown under light in a thin, transparent lollipop. Radioactive carbon-14 in the form of hydrogen carbonate is injected at intervals, seconds to minutes. Samples of the photosynthesizing algae are dropped into the hot methanol to stop chemical reactions instantly, so like taking a snapshot in time. The compounds of the algae are separated by a two-way chromatography. Okay, two-way chromatogram. The radioactive compounds are identified. So, if you think about it, it ended up looking like this. So, carbon dioxide is what he noticed first. That was the radioactive stuff. That's what they saw first. They didn't see that. They saw that. Then they saw that plus that. Then they saw that plus that plus that. Then they saw that plus that plus that plus that. And then, after that, it was just those all the time. And that's how they came up with this cycle. Okay? So, that is how science works. And he got the Nobel Prize for that, because of one of the most important reactions happening on Earth. Now, typical exam question you can get is based on some evidence here, so you've got some time. Suddenly the carbon dioxide is decreased, you're not giving any carbon dioxide anymore. Look at this. There's RUBP. So this is under high concentration of carbon dioxide here. Quantities of RUBP and GP. Well, what can you tell? The green line is your RUBP, the red line is your GP. Well, what can you tell about the quantities? And then what happens when you suddenly stop giving it carbon dioxide? You need to be able to interpret this. This is a typical exam question that comes up, this kind of picture about photosynthesis. Well, what you should notice at the beginning, if we just give these arbitrary units, okay, it's because there's no actual units here, but look, if I've got 1 box, 2 boxes here, so that's about 2 boxes. So it's 2 boxes and 1 box, so it's like, let's say 21 boxes. Whereas this one is 20, 40, 42 boxes. So 21, 42, well, what do you notice? Well, the GP is twice as much as your RUBP. Is that right? Well, yeah, it is right, because remember, RUPP, there's only one of those, one 5-carbon compound. But then when you split it to make GP, you split it so that the RUBP gets split 2 ways, you end up with twice the amount of GP. Right? Because that 5-carbon compound becomes a 6-carbon compound, then it gets split into 2 lots of 3. So the quantity, it's just the amount of it, is twice the amount of RUBP. And we've got lots of carbon dioxide, happy days, the quantities stay in proportion. You suddenly stop giving it carbon dioxide. Well, if you're not giving it any carbon dioxide, RUBP can't turn into GP. So what happens to RUBP? You're stopping the assembly line. RUBP starts to rise because you just sort of put a line in the way, and it's like a building, like a flood of it building up. There's no carbon dioxide joining onto it to turn it into that. So it starts to rise. Well, what happens to GP? GP starts to go down. Okay, it's kind of obvious because if RUBP isn't being converted into GP. Okay, so typical exam question. So this is a summary then over our last 2 lessons. We looked at light dependent reaction and the light independent reaction. There's your light dependent. On this side is your carbon cycle, the light independent reaction. So this summarizes everything that's going on. It's a sort of simplified diagram here. Light hits the chlorophyll molecule, light hits the water, photolysis gets split. This energy from the electrons makes ATP. That ATP can be used as active transport to pump hydrogen ions, remember. Those hydrogen ions then eventually get mopped up by NADP to become reduced NADPH. Those electrons also get mopped up by reduced NADP to make that neutral. The light hits the chlorophyll molecule, the electron gets raised by the energy levels, gets excited, but as it comes down the energy levels again, we're getting ATP being produced as well. So the products of light dependent reaction is ATP, NADPH or reduced NADP, and then they are used for our Calvin cycle. So we got your carbon dioxide joining your RUBP. RUBP becomes 2 lots of GP, and then those with the use of your ATP and your reduced NADP, the GP becomes reduced to make TP. The TP then gets turned into glucose. The TP also gets reconverted back into RUBP with the help of ATP, and therefore this cycle can happen over and over again. That's a summary of all the photosynthesis biochemistry. Next time on My Smart Learning, we're going to be looking at limiting factors, which you've done at GCSE, but it's going into probably a little bit more depth. So hopefully you've learned something about photosynthesis today, and I hope you're keeping well. Can you make sure you do all your summary questions, your notes, your exam style questions, do the stuff that Miss Martin is setting. From what I hear, you lucky lot, she's going to be doing some Zoom lessons, I think, in this week or next week, catching up with you guys via Zoom. I may follow suit if it works out, looks like it's being successful. So take care, and oh, I forgot, something you can't buy in the shops. I haven't given your virtual high five, so here's your virtual high five from me.